direction
Yeah, I
you were
think
going.
it just kind of shrunk up some, but
Yeah.
I think here we can zoom in closer. Isn't that it right there?
I think it's underneath it.
Yeah,
I think
I think that is another sponge. Yeah, and I think we're underneath that one.
Yeah. So if you zoom in right there, you can kind of see a depression. But I think the opening's on the side we can't see. Yeah.
Yeah. Yeah, kind of like what's on the screen now. I think it's that spicule blob.
All right. You can go to the forward box anywhere.
I could put it in the side if you'd rather.
Yeah, you could do that. It'll fit in one of the small boxes.
Yeah. Oh, you want?
Sure.
Okay.
Paul, can we see the starboard bio boxes, please?
Hold on 1 second now.
Got it. Oh, yeah, you're doing brow cam. Need an octopus sitting in your chair. Sir?
All right, sample tray out.
Yep. All right, which one? Oh, come out more.
So you're going
Which
B or C, the two inside interior compartment. All right, it's on B.
.072, Steve?
Correct.
Fab, thank you very much. No, it's on there.
Challenge complete.
Woo.
Ooh. With extra sponge.
Yeah.
All right. Niskin complete, sample complete. What's next, Debbie?
Good job.
What?
So we just finished a transect, correct? Abby, would you like to poke around some more here and explore?
Yeah, I mean, there's so many different types of sponges here. I would love to, I mean, if we could make our way in sort of the general direction, but I think it would be great to just sort of grab some zooms on things. Sometimes it's not obvious if we've taken a few samples here, but just to sort of make sure we've sampled the different types of sponges that we're seeing in this area.
Roger.
Okay, so that sounds good. Let's proceed.
Alrighty, we'll just
do a junk zoom there, Al? Oh, not that much.
Oh, junk zoom.
Junk zoom.
I thought you said jump zoom. I'm sorry.
Junk.
Bob, are you good with making a ship move?
Yeah.
Starting? Perfect. Bridge nav.
I thought we had a junk zoom.
30 meters, 343.
That's
isn't that the term?
That's a thing now.
Yeah.
We made it up, you and I.
Three what?
343.
343.
So science, when you do see something you want to see, just let us know quickly if it's a quick zoom or a stop the ship to sample or take longer zooms, please.
Roger.
Yep.
We're going to be brought into the current that way. Huh.
Oh goodness, yeah, I can see lots of them kind of everywhere now. Someone on the AUV team calls them the monk sponge because they've got that fringe of hair just around the edge.
Which one is that, Abby?
The thin ear.
Oh,
Yeah. In the nest. So I know they've collected some faria. That's a glass sponge we're seeing on the right coming up that kind of looks fluffy and lacy. We've collected some of this thick plate sponge. And I know Tom collected this yellow one that seems to kind of grow in like vertical ridges, sometimes on stalks, that is a type of Mycale. They've also collected this big globular sponge we're coming over now. And there's some yellow, fine yellow tubes as well that have been collected. A big glass sponge coming up.
Do a quick zoom there, Al.
Quick zoom.
with a little thorny head next to it.
Yeah, turn lasers on.
I was just going to say, could we have the lasers? Thanks.
The current's ripping.
Went out a little bit. All right, coming out wide.
Lots of food in the water, wouldn't you say, Abby?
Yeah,
One of the reasons why these sponges are here.
Yeah, and the high current especially. So sponges filter feed and they do create their own kind of current internally. They have these specialized cells that kind of waft water through their bodies and extract little particles as they feed. But when they're in high current, it sort of helps them by bringing food to the sponges and facilitates that feeding process. So yeah, you tend to find lots of sponges, particularly in areas with high currents like this.
And just a reminder for those watching, those two green dots are laser dots from laser beams, and they are 10 centimeters apart, which is about 4 inches. And they help us size things as we go along, determine the sizes of, estimate the sizes of fishes, corals, and sponges, and so forth. And we're at about 5. Yeah, go ahead.
Oh no, I was just going to go on about how we're near the footprint, which is kind of an offshore bank.
Yes, go on. I was just about to tell people where we are.
Yeah, and it's a site that certainly with the AUV we've been to before, and I think this is also where there was a comparison study done in 2011 where they used a submarine, an AUV, and an ROV to survey the same area to get estimates of fish biomass. So
yeah,
we do have AUV imagery here.
Yeah, we did that that comparison between the vehicles. The AUV may have been down here, but the the other two vehicles, or at least the dual deep worker manned sub that we used, we were higher up on the actual
On the
footprint
top.
bank. This is up 400 meters and shallower. We're at about 500 meters here on the sort of a deeper extension of that footprint bank. So I've never seen this type.
I mean, no, and I think we must be in a different site. Well, the AUV tends to go, I suppose, for the lower relief areas anyway, but certainly when I was looking at the data, I think we were in a lot of mud.
Yeah.
How far do you want to go?
You can zoom in more.
Oh.
Do you recognize that coral dye?
Let's look at the chat.
It's thick, so it makes me think of bubble gums, but I'm not.
Yeah, bubble gums or Stephen Cairnside coral.
Do you know what this
left the
sponge
room.
would be?
Did anybody notice the
I
side
do
of that
not.
rock? Cross section there.
You want to look at this longer, or I need to kind of put my foot in the dirt?
Can we pause for just a moment? Would that be okay to pause for a second? I was going to look up to see if it's on our list.
It says if it is paragorgia, then all of them are kind of desirable.
Should we collect it then? I just wanted to see, make sure. Kurt says it looks like paragorgia Stephen Cairnside, but hard to tell. That was my thought. I think we've collected one earlier, but perhaps let's take a piece of it now, if you agree, Abby.
I can look quickly back through.
We did collect a piece of what we thought was that species yesterday, but
okay.
It's up to you.
I think because we're doing the ground truthing as well for Matt Potey, we should collect.
Okay, let's do it then. Thanks.
Okay, what are we doing with this thing? Where's it going? Zoom out.
I think all we need is a small branch, right? Shoreside?
Yes.
Yeah.
Yes.
So is it a snip and slurp?
I think that'll work. Not too dense. Yep.
Gonna flush now.
Okay.
Is there a jar that we're going for?
4 is next.
Oh.
reminding me of a gangster movie. Let's figure it out.
You'll notice all the polyps retracted while we were disturbing this coral, trying to collect a piece.
So for the past 2 years, I think there's been a cross-shelf modeling project. It's sort of carried out by BOEM, the Bureau of Ocean Energy Management, and Matthew Potey is the main modeler. And one of the things we've been doing on the express cruises and now with the Nautilus is going to some of the sites with high predicted sponge and coral diversity and actually going and looking on the ground and ground truthing that model. So this is really, really useful.
Ready for section?
Yep. Ah,
Yes, and those predictive models, oops, can we get it? Is it
Seems
lost
like
forever?
I think it, hopefully it didn't blow away in the wind here.
Looks like it dropped pretty straight.
Yeah.
Where'd it go?
there's one piece in sample jar 4 now. It looks like there were 2 pieces. One
Is that-
small one
yeah,
blew
there's-
away,
there's
and the big
actually-
one is in 4.
Okay, that's fine then.
Okay, so
You got
one
enough?
made it in.
We're good.
That's
Chip it.
okay, great.
Could,
Whew. Abby, you've got
Nice.
another sponge you want to have a look at. Good
yes,
job, you
please.
guys.
It's actually, thank you. It's actually just underneath that coral, if we're still good in this position.
Sponge just to the right of
Yep.
the sponge?
It's
Or, I mean, of the coral we
to-
just got? Yeah?
Yeah, correct. To the right and just slightly down from it. We could possibly have a zoom on that and potentially take a piece.
Okay.
I'm seeing Ryzax and Elegadis, I think, 2 die on the left, but that one is not. I think that one's pretty distinctive. Do not need a piece.
Okay.
Steve, the coral is 073.
Correct.
Thank you.
I'm going to mute here for a moment to type so you don't hear my typing.
You know, do me a favor, please, and don't mute. I'd like to see what the effect is now that you've got a new configuration.
Okay.
Okay. You want to zoom in now?
That's what I live for. Yeah, we do hear you. Not as loud as before.
Okay.
Thank you.
Lovely zoom, thank you.
You want lasers off? Lasers off?
Yep.
Oh, nice.
So, do we want this or?
Yes, please.
I'm pushing
Okay.
HS.
Did you get it with
It
the
looks like
lasers on? I wasn't paying attention.
Yes.
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 Okay.
So where's this gonna go?
Is it slurpable? Shoreside?
It looks like it would be quite soft, so I think it probably is slurpable.
Okay. Jar 5?
Abby, tell us what you're thinking about this sponge.
I think it's a demo sponge. We've been in the AUV imagery when we've been coming across sponges like this. I think this is one that we've just sort of been calling a white or lighter Mycale. But we do not have a sample to sort of confirm that.
And while we're collecting, can you tell us a little more about your work with sponges, Abby, just for those out there listening that don't know?
Well, so when I first started working as a contractor at the Northwest Fisheries Science Centre, there was some interesting data about some really spongy areas off the Olympic Peninsula off the coast of Washington, where they knew that there were high densities of these potentially reef-building glass sponges. But up to now, glass sponges were only kind of something that had been, they used to be extremely common, and then we thought that they had disappeared, and then they were kind of rediscovered in the 80s off the coast of Canada. So in certain areas, these kind of very unusual sponge reefs form, where you have just sponges growing on the dead skeletons of other sponges, and they form these really, really unique habitats. So my boss, Liz Clark, wanted to go and investigate whether this could also be happening off the coast of Washington. So used a combination of AUV imagery to take pictures of the sponges. The Nautilus went and visited that area and took some samples of the species that were present there, and also did some sub-bottom profiling to kind of ascertain whether we were looking at the interesting features on the ground, to find out whether they were kind of solid, made of rock, or whether they might be these sponge reefs. And they have a very interesting kind of acoustic profile. When you see the Canadian reefs, it kind of almost looks like clouds on the substrate. They can get up to 19 meters tall in some areas. Yeah, really incredible. When I came along, my background is sponge ecology, but all on coral reefs and shallow waters in Indonesia. So it was quite a change of environment for me. But that was the first job that I was doing with Liz. This is like a lovely sample, by the way. Can't take my eyes off it. Look at that. Perfect.
Fantastic.
So we took all that data, we used the sub-bottom profiling, the presence of particular species, which we know can form these reefs, and then the images on the AUV to look at densities.
Wait a second, wait. I don't want to blow this.
They're about to slurp.
Okay.
Drum roll.
All right.
Could it even go in a bio box, or do you think it's too floaty?
Oh, well, we can put it up front if you want to try and keep it.
You can put it up front.
Yeah, to try to keep it from getting mangled.
It's just so nice.
Hopefully it stays that way.
Yeah, it does look really floaty.
Well, it has a little piece of rock on it. Oh.
Okay, yeah.
Up front's a lot safer because you don't get the thruster wash like you get on the side.
Oh, got it. Yeah.
Okay,
so
box
try
out.
Yep.
This is what we're talking about
side?
when we refer to the bio box. It's a series of compartments that we put the samples in, or that the pilots put samples in, I should say. And then these will get processed.
It's not
on
overly
board the
floaty.
ship.
Looks like it'll stay in there.
Oh,
hooray! Fab. Thank you. Fantastic.
Good job.
And I promised this would be the last one, but if we could also zoom on the white sponge, just to the left.
This is a veritable
Never go to the
cornucopia
grocery
for
store
Abby.
hungry.
Yeah.
Yeah. It looks good because it looks like it's actually got some damage already. So this would be these particular.
A good opportunity. Yeah.
Yeah.
Go for it. And we did the Niskin already, right? Yeah.
Lasers on?
Yeah.
There they are.
All right, good captures.
Okay. Lasers, well, I guess it doesn't matter. It's all right. So, what are we doing with this one?
So this would be one that will be much kind of tougher than the last sponge. So probably more of a grab.
Okay.
Yeah. And I think it will be a sinker.
Okay.
Do we have cutters this time or not? Just wondering if they're available.
Yeah, we do.
Yeah. Yeah, we
So
switched.
Abby, that option is available as well.
Yeah, I mean, it shouldn't be, it's not as tough as a coral, that's for sure. It's they have sort of fiberglassy type texture. And there are a lot of different species, and they also have, so there are different spicules on the outer and inner surface. And these particular ones, they have little, they look like little umbrellas that stick out of the outer surface. And on some of them, they have thorns, and some of them, they're smooth. And you can only really tell when you get them under kind of a microscope.
So, are we trying to snip it? What are we doing with it?
So if you could get a piece from the top and some of the body, and just kind of rip a piece off.
Well, I can cut it. We have
Okay,
the
yes,
blade,
yep.
so.
Yeah, the cutters are available, Abby.
Okay.
The blade.
Can we zoom in more now? Need more juice here.
If the whole thing comes up and it can go in a bio box, then so be it. We do not have we don't have any of these sponges from this particular site.
Looks like the whole thing might be coming up.
Yeah, I think.
Yeah.
Well, I got a piece of it, but is that okay? Is that enough?
That is, yeah, that's fantastic.
And where's this going?
You want to go
Slurp?
forward B or slurp? Your call.
My call.
Yeah.
Not my sample.
You think it's gonna clog it up?
So this would be the same kind of sponge that did clog up the slurp yesterday.
Yeah.
Let's
go forward B.
Forward B, all right.
So that leaves us one more open compartment for sponge on the starboard box shore side, and then we got three more slurps.
Yeah.
Just kind of.
Thank you.
Don't boosh it out. Just want to keep the goodies inside. That's good.
So Lisa was suggesting we should probably be going into some more coral territory later on in the dive. So any corals can double up with sponges.
Sure.
So we know we've got plenty of capacity for that. There's another sponge that I'm keeping an eye out for, which would be a slurpable.
Get in there.
Whoop.
Perfect. Oh.
Down there. Good job.
Thank you. That was fantastic.
Abby, is there a name for that, or are we just calling it glass sponge for now?
I tend to lump them just as kind of lysocene glass sponges. So they're glass sponges in a order where they have spicules that are not fused together.
Okay.
So I can put that in the science chat.
Yeah, that'd be great.
Okay.
Okay. Abby, if you're okay to proceed, we can make our way toward the next waypoint.
That sounds fab.
Jump
Okay.
zoom now.
Roger, we will continue towards waypoint AD.
That was 3-4 something.
343, please. Bridge nav. 30 meters, 343. Quite the Sponderland.
Lovely, isn't it? Pretty. And quite
Just, yeah.
varied.
Call them
So
a
Abby,
sponge garden.
yeah, feel free to speak up if there's something else we should stop and zoom in on or collect.
Well, I'm already seeing, so this chap coming up here that kind of looks like a little heterocone. There's slightly different morphology from the ones that we see off the coast of Washington is also on the list. So if we're not moving too quickly, if we could have a zoom on the one that kind of looks like it's got these lots of finger like projections, but they seem to be fairly common, so it doesn't have to be this chap.
Let's see, we're talking about over there?
Yep.
Okay, oh boy, the current's ripping.
You want to move on and find another, or you think we can zoom on this one?
Just gotta get where I'm not broadside to the current. It's hard to stay put when you're got a sheet of plywood in the wind there.
Yeah.
It's not a very nice thing to say about Herc.
Or like a box truck going over a bridge
Yeah.
in the high wind.
I think that's better. At least it's a machine.
Yeah,
Not
better than a
piece
sheet of plywood.
Hey Sam, can you do my pan and tilt here while I drive?
Sure. Where is it?
That other stick, or
This one?
somebody do it.
Oh.
I'm muting because I'm
All right,
typing.
there you go. I don't know if this is gonna work.
Okay. Yeah. Apparently it's a common sponge, so we can continue if it's too hard to set here.
Well, no,
Yep.
we can zoom in. You're just not gonna get it quick. Can you zoom in now? I'll just try and. It's just precarious here. Can you zoom in more? There, get your shot.
Thank you, Fab.
Perfect.
Perfect.
Okay. Good capture.
Okay.
All right.
Nav, can you add 75 to that list of waypoints there?
Okay, I think you've got to race ahead here a little bit.
Okay. Racing.
Bridge nav.
Junk zoom out.
30 meters, bearing 343.
Oh, I think we're racing different ways.
What? 343. Oh, because I'm getting blown in the current here. Yeah, it's really ripping. All right, I gotta turn into it.
Roger.
I'm gonna have to do more of a crab here.
More of these little Amphiptilum sea pens, I think. A little
You gotta be like a
curved
scuba
over.
diver and you stay close to the bottom. And,
Would it help moving the ship over to the east a little bit?
Use the claw to.
claw our way across.
Yeah.
I want to get video of that.
I've seen divers hanging on for dear life, and they look like kind of pegs on little laundry on a line. Trying not to fly away.
I think that was a Dover sole. I'm not sure about that flatfish.
Are we collecting sea pens for a project, Di? I see they are on the list.
It's easier to look at.
It looks like it's on an old sponge too.
Yeah, that's
Yeah,
zoom in on that?
sponge
zoom in
rubble.
on that.
Oh, yeah, this is perfect because this shows you, so where they do have sponge reefs, this is kind of the process. You have a dead sponge skeleton, and then you have new sponges growing on it. You also have one of those, the near or the monk sponge.
I'm going to look at what's been collected so far, Abby, while we're zooming in here. But they were going through a large sea pen field with lots of Anthoptilum and Holypterus before. So I'm guessing if they were going to collect, they would have there, but I will check.
Okay, lasers off.
All right. Perfect.
I don't see sea pens having been collected. Now I'm going to look at the list, the wish list.
Good captures on this scene.
I do see Anthoptilum on the wish list for collection.
Yep.
Maybe when they were going through the sea pen field before they were on transect. I can't quite remember. I tuned in, a little late.
If anyone's chiming in on the chat, but otherwise I would be tempted to collect just because they are on the list and we have not collected any.
Right. No one's chiming in, but I think Meredith has. Oh, Gary Williams just joined in. Hello, Gary. I think Meredith has left for the time being. She was on the 12 to 4 shift, so I imagine she might be catching a wink or two. Yeah, I agree. I think we should collect one after we're done with this Zoom, if there are still some in the vicinity.
Okay, zoom out now. And what are we doing with this one?
Were you interested in collecting the sponge or just having a look, Abby? What was your thought here?
I would like to slurp one of the yellow sponges with all the little projections. It looks like a kind of heterocone. They're very
zoom in.
One of the two big ones on the right. They're very fragile, okay. So it should be just to slurp a little piece.
Oh, okay.
This illustrator's up on 3, Steve.
Not the little tiny yellow ones. Is it?
Not the tiny yellow.
No, they look orange from our camera.
Yeah.
Okay, zoom out.
Is our ship move complete, Sam?
The ship's holding position.
Okay.
Yes, we're definitely getting a yes for collecting an Anthoptilum.
Okay, great.
From Gary Williams.
It's always nice to just get that confirmation.
He says the Anthoptilla may be a rock pen and a possibly undescribed species. Please collect whole body if possible. Roger that.
Oh, wait, a whole body?
Oh no, not the sponge.
We're talking,
Sorry.
not the sponge.
No.
Sorry. The next collection.
You're thinking ahead.
Sorry, we're getting ahead of ourselves.
And what's the species name for the sponge?
Definitely heterochord.
Okay, zoom in.
Possibly heterocone calyx. It's a species we see right up and down the coast, but with slightly different morphologies in different locations.
Ready for suction?
Yep.
Going in jar 5.
Jar 5.
Okay, lovely. Oh, no, wait. Two.
Zoom out, Al.
Got
At
a
least
small piece.
one piece has made it in, 2. Looks like 3 different pieces now.
Thank you. Yeah, that's perfect for that. Nicely done.
That's what I do.
Bob, can we do a DVL reset here?
Yep.
You've done this a couple of times now, haven't you?
Couple.
All right, I'm going to go over to flush and
All right.
Okay.
What's next, Shoreside?
Our next guest, I'll call it, not victim, is
Welcome aboard, guest.
Patient.
a patient, is one of these little purpley red sea pens over to the right.
Hey, zoom in.
Correct?
One of the big ones.
Guys.
There's some on the left too. They seem to be all around here.
Yeah. And they'd like us to collect the whole body,
Okay.
the whole colony, I should say.
So, and where's that gonna go? Is that a slurp or is that a?
I think it's a slurp.
Okay.
Let's see if we're getting any commentary on slurp versus. We will, Gary will need the base, Meredith says. They've got a little anchoring base in the sediment. It may be attached to something hard.
You want to poke at it and see what it looks like?
And a good zoom too, I think, if it's maybe a new species.
Okay.
Yeah.
you want the lasers on?
Thanks for remembering that.
Zoom out
Lasers on, yes, please.
I may need to reposition here because I can't.
Sure.
Okay.
There's several to choose from, so whatever works. Meredith says if it is the rock pen they think it is, it could be, the base could be like a suction cup. And they're saying to try to find one that's clearly attached to a rock if we can. So that changes things a bit.
Can you zoom in now?
Thanks for clarifying that, Kurt. Do we have the lasers on? I can't
Yeah, I
tell.
think we're just zoomed in too much.
Oh, okay. Yeah, I see him.
Hey, zoom out, Al. Zoom in.
I'm going to type to our chat advisor, so excuse my typing.
Yeah, I think this one looks like a suction cup from the view that we have.
So you think it's on a rock?
No, I don't know if it's on a rock, but at least not one you can grab. You might be able to get under it and dig the claw in a little bit.
I could poke at it and see. Zoom out, Al. Zoom in.
Di's just asking for some more guidance on the science chat about whether they have a preference for which, yeah. Oh, that's a good shot. You should be able to see.
Yeah, that's a great shot.
Get more zoom out?
No, not really.
That's it.
That's the end of the lens.
All right, good captures.
Well, I'm not, oh, here's Meredith, okay.
I don't see any that are obviously on a rock, but
yeah.
Zoom out, Al.
We're waiting just a sec to see if we hear back from Gary. He's on the chat.
Zoom in up there. Just kind of look around, I guess.
Well, that's a nice view of the base of that one, but it's not on the rock.
That's a really good size one there. That 2?
Looks yeah, like it almost looks like a second branch.
Oh,
well,
no, they just separated. Hard to tell.
Meredith says, can you zoom in on that one in the lasers? Or are we all the way in?
Yeah, I think that one's kind of far away. We can
make it. Now I'm at the end.
Oops.
Okay.
I'll zoom out to make that easier.
Nice shot of the short spine thorny head.
That's it.
Gary says possibly attached to a stone beneath the sediment. And Meredith is asking Gary if it would be worth trying to scoop one up.
We don't have the scoop aboard, but I could try and cradle it.
Okay.
Yeah, potentially how deep would the peduncle go into the sediment? I guess it depends on how deep the rock is.
So can it live up forward with the other samples, or?
Yeah, I think it can.
Okay.
Yeah.
Zoom out, Al.
So we're just waiting for a little more feedback on the chat. Thanks. So we don't have the scoop, you said, but we have some other way of
we could
kind of getting underneath it, you said?
Yeah, we can cradle it in the fingers.
Okay, Gary says the peduncle should be right below the surface, might be able to dig it up with the claw. So let's give that a try.
Okay.
Thanks, Gary and Meredith. Okay, so
Current is whipping.
Like that moved it a bit.
Can you zoom in more? Tell what's going on there.
Maybe from the other side. That's a delicate operation. It's fascinating to watch how precise you can be.
It takes a lot of practice to get like that.
I don't think it's on a rock at all.
Okay.
Doesn't appear to be.
Well, I think we still want to try to get the whole thing, and I think you're on your way to doing that.
Yeah.
Oop, there it is.
Can you give me a tilt down? Ah, come here. Can you deal with my pin and tilt there?
You saw me looking up, didn't you? Yeah.
Ugh. I want to cooperate here. Oop, don't go anywhere. You just tilt me. Just keep the thing in view.
Yeah.
I think we're at the
Yeah.
hand limits.
Try this. Come on.
Either one of the forward boxes, Bob.
Wow.
It's not in a box yet.
Sorry.
Sound like counting your corals before they hatch.
Holding my breath.
We ready for box out?
Yeah. Stay.
Woo.
Wow.
Woo. Woohoo! Yay!
Great job.
Wow. Well done.
Whew. It's like the most dramatic moment of the morning.
Need to wipe the brow of her. And now would be a good time, I think, since we've got maybe a new crowd joining us, for everybody to just introduce themselves really quick for my sake too.
We can do that.
All right.
Great.
Start with you, Sam.
Sure. Samantha Wishneck, navigator, and Steve 077. Thanks.
Yeah, I'm
Okay.
Steve 077.
And
007.
Bob Waters, I'm the Herc pilot.
Paul Glick, I'm the Argus pilot.
And Al Santos, video engineer on the watch. And
And
Mr. 077.
Yep, and I'm Steve 077. Anthopelum CPen, and I'm in the data logger chair.
He's in all the science chairs today.
I'm in all of them. I get the whole back row.
How can you give me a
lost
jump
you guys.
zoom? Oh, I gotta, here, wait. Let me push forward again. Okay, now jump zoom.
Shoreside, before you introduce yourself, do you want to keep moving forward?
Yeah, we can move forward while we chat.
Roger.
So we cut out after Bob Waters, who I guess is not my relative, since you're a Waters with one T and I'm a Waters with two T's.
Ah. Actually, my father's
maybe
name was Rainwaters, and he changed it. So.
Really?
Yeah.
Wow.
Yeah. Which
Some
is
of
a
my-
British
some
name.
of my okay. Rainwaters. That's pretty good. I've been nicknamed Muddy, Shallow.
Oh,
and
yeah.
let's see, I've forgotten some of the other ones.
Yep.
Okay, so Bob, you're the Herc pilot, and then it cut out after that.
Oh.
And of course, we could look this up, but it's nice to hear your voices and just say hi.
Ah.
Yeah. I'm Paul Glick, I'm the Argus pilot.
And this is Al Santos.
Hi, Paul. Hi.